This is episode 73 of the Immunology podcast, T-cell receptors with Dr. James Allison. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology podcast, where we have conversations with immunologists. The Immunology podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. James Allison from MD Anderson Center on the podcast to talk about his research on T-cell receptors and immune checkpoint blockade. We've also got our usual roundup of recent highlights of immunology news coming up. But first,
Immunology 2024 will feature 8 major symposia, each focusing on a specialized area of immunology research and featuring multiple presenters. Topics include peripheral neuroimmune interactions, cell death and immunity, innate immune memory, aging, obesity and immune responses, immunotherapy, myeloid cells, mucosal immunity, and immunity to emerging pathogens. Don't miss this all-immunity gathering in Chicago, Illinois, on May 3rd to May 7th. Visit immunology2024.aai.org for more information.
well, we have a big guest today.
We do.
I know you were practically bouncing out of your chair for this.
Oh, absolutely. Yeah, I'm so excited. So looking forward to sharing this conversation we had with the one, the only, the father of immunotherapy, James Allison from MD Anderson Cancer Center, Nobel Prize winner in 2018 for his amazing work on bringing checkpoint therapy to the clinic. For those in the know, the anti-CTLA-4 therapy was envisioned and pioneered by his lab, and that really opened the floodgates to checkpoint inhibition and completely, I think, changed the landscape of treatment of melanoma, particularly lung cancer, and so many other tumor types are being tested nowadays with so many amazing results.
So what do you think is going to win next in immunology?
It's tough because a lot of the latest things in immunology is hard to pinpoint them to one person. But I guess the mRNA vaccines have already been awarded last year, so that would have been an easy guess.
How about cell therapy with Carl June or someone?
That one I like. Yeah. Maybe CAR T-cell therapy, because also, especially now that there's also this huge growth in CAR T-cell therapy for autoimmunity. So we've covered actually a couple of those recent papers. So it looks like CAR T therapy is a lot more useful than we initially thought. So yeah, I would like that. I would like to see something like Carjun winning for CAR T cell therapy, or other people that worked in the early understanding of T cell biology. So I think there's many options. What do you think? Something with microbiome? Is there anybody?
Post microbiome stuff could eventually get there. I don't think it's time yet. You need to have it fix more diseases. And they tend not to do it posthumously. So
That is true. Yeah.
I don't know if the people who pioneered it will be around when it's time for that.
We'll see. I mean, now we have to wait until October this year for the upcoming winnings. So, but we don't know what's going to be in.
There'll be something else this time. Immunology had its turn last time.
Yeah, I think so too. But well, where it's always the turn of immunology every time, all the time.
Right here, right now.
Right here, right now. So,
right. Well, I'm going to tell you about antibiotics.
Antibiotics. Okay. I think we've been doing this for a while now. You have a couple of...
This is the second time. Two is not a while.
Okay.
I'm young enough that maybe 2 is a while.
Okay. So what's the story?
So this is a related paper to what we talked about last time, but it's a big deal in science. It is an antibiotic pre-organized for ribosomal binding overcomes antimicrobial resistance. First authors are Kelvin J. Y. Wu and Ben I.C. Tresco. Last author is Andrew G. Myers. So this is almost an organic chemistry paper, but it's also a microbiology paper. So they looked at macrolide-based antibiotics and used structure function work to see how they can make it better. So fundamentally, we have some really potent antibiotics, but they develop resistance, or bacteria do. to these macrolides, like lincosamide and streptogramin B, because these inhibit the ribosome by binding there and preventing, the ribosome from functioning. But then a methyl group gets put on the ribosome that blocks their binding. And there's a couple different methyl groups or acetyl methyl groups that get put on in different bacteria or dimethylation, and it kills the antibiotic. Those genes then transmit because they have fit to test capacity, and so you get resistance. So they used a kind of structural understanding of this to try to see if they could make something bind tighter or slightly differently, and how to have less freedom of movement in the binding pocket. And they did some fancy organic chemistry, which is impossible to describe here. But they basically built out the macrolide ring on the other side to make it more stable with some bridging groups using some pretty sophisticated organic chemistry. And then lo and behold, through combinations of crystal structures and antibiotic, so that they threw it with bugs, right? And showed that it kills antimicrobial resistant strains. E. coli, E. efficalis, Staph aureus. It worked incredibly well on everything. Pseudomonas, Klebsiella, C. diff, gonorrhea. So it's fabulous as an antibiotic. And they try to figure out why. And it actually, because of how it works, it pushes against those methyl groups and slots in next to it. So if it's methylated, it causes a twist. It binds to the ribosome anyway and twists the ribosome, further making it not work, the ribosome not work. And there was no way they could have predicted that ahead of time, but they got it to work. And this antibiotic is called crestomycin. And they do a good job claiming we don't even know it's maximally optimized, but they don't see any toxicity on early screens of cell cultures in animals. And it looks like it could be the next antibiotic to come out to fight, multi-drug resistant bacteria, the escape pathogens. But the paper's only 6 pages with 5 figures. And it is super sophisticated structural biology, or really structural organic chemistry with some X-ray crystallography and a little microbial inhibition assay. But they're finally making new antibiotics.
About time. That will give a Nobel Prize to that, because antimicrobial resistance is definitely a looming catastrophe coming towards us. I have a question. I always when there are these antibiotics that affect RNAs, what is the risk of targeting mitochondrial components?
This is ribosome, right? So these are into bacterial ribosome, which are sufficiently different from human ribosomes as to not have real toxicity.
Yeah. Okay.
Right. This isn't like that. That's why you have toxicity with yeast antibacterials, because yeast is a eukaryote. But
yes.
prokaryotic ribosomes are, it's a 16S, not an 18S subunit. And so you can make very specific antibiotics to bacterial ribosomes. That's why we do it.
So what's up next? How do you test this? How do you can you bring this to the clinic ASAP, or how exactly does it work?
I mean, it's kind of up to them, but if they had the money, they could take this in the clinic immediately.
Yeah.
The GMP manufacture it or manufacture it to clinical standards instead of in a organic chemistry lab. You'd have to scale up the manufacturing. So they show 100-gram synthesis and stuff, which is a good start. And then it's time to treat. So it's really scale it to a clinical grade, clinical trial grade manufacturing and putting people to clinical trial, which of course is not just a little thing, but like if some company came along and wanted it, that's what they do.
Well, I hope that works out. So for my first story of the day, I have a very interesting story about, and forgive me for the topic, T-regs. It's always T-regs. They're always everywhere. But I love this. I think this story is very interesting, and it's an overall arching theme that I find very interesting. This is about visceral adipose tissue infiltrating T-regs. The paper is called Two Regulatory T cell Population in the Visceral Adipose Tissue Shape Systemic Metabolism, published in Nature Immunology on Valentine's Day. First authors are Santiago Valle Torres and Kevin Mann from the labs of Ajit Kumar Basantakumar and Axel Callis from Peter Doherty Institute and the University of Melbourne, respectively, in Australia. So it's been known for a while that you find, particularly, a lot of these experiments are mostly in mice, but many of these uh, also applies to the human population. It has been already known for a while that there is a population of regulatory T cells that live in the adipose tissue, particularly this visceral adipose tissue that's in kind of in the belly area. And this was pioneered by uh, uh, people at the lab of Diane Mattis, who's well known for her T-reg studies, first Marcus Feuerter, and this been known for like 10 years now. And what has been observed that this T-regs are very particular. They have very specific transcription factors associated with their survival in the fat, particular PPR gamma. This is a transcription factor that is activated by certain lipids and is usually associated with adipose tissue. And these T-regs, they don't only exist, but they seem important, and their presence seems important to maintain homeostasis, kind of metabolic fitness in these mice. And particularly, they seem involved in sustaining glucose tolerance and glucose metabolism systemically in mice, which I always find kind of, well, what's the word? Remarkable that there's this population of T-regs in the adipose tissue, but if you remove them, these mice do lose some glucose tolerance. They don't regulate glucose anymore so well anymore. So they do seem to play a role. But what is also interesting is that these T-regs are found, they've been were originally characterized and are really characteristic of the fat of male mice. Usually, male mice have, when they get older, they have more fat, and they do seem. It does seem that age, and also if you have a high fat diet, one of the things that you observe is a loss of this Treg population, this PPR gamma positive, also ST2 positive population, and that this correlates or even causes dysregulation of glucose metabolism. Interestingly, this is not exactly the same in females. Females seem to have a different their T-regs in their fat, their visceral fats have different characteristics, and that has been shown that they seem transcriptomically different. They have different markers, and it's so I've been following a little bit this topic. There seems to be an unclear, but recent publications show that one of the cytokines that are important for the establishment of this Treg population is IL-33. So we already established PPR gamma, IL-33, and the absence of IL-33 affects the presence of these Tregs and in turn, metabolic dysregulation. And this is particularly in male mice. So what they do in this study, they look a more systematic view between males and females and young and old mice and mice subjected to a high fat diet. And what they see that is indeed these they can actually kind of take apart two distinct different Treg population that can be found in the adipose tissue of mice, and they seem to be preferentially present in either males or females. And this PPR gamma positive IL-33 responding Treg population seems to be characteristic of males, and apparently this is related to the expression of IL-33 by mesenchymal cells in males specifically. So what they do single cell RNA-seq of Tregs infiltrating, young, lean female or male mice, and they do clustering, and they show that there's indeed two different populations, and they identify a new kind of previously not really characterized population of cells that are enriched in Th1 transcripts. So particularly CXCR3 and TBET, which is, this transcription factor associated with Th1 cells. Also, CXCR3 is also associated preferentially with Th1 cells, and the expression is driven by, to some extent, by TBET. And this is, and they see that whereas male mice are more likely to express this PPR gamma, IL-33 responding T-regs, females seem to tend to express less T-regs, but more of this other phenotype. And so they look into these two populations, and they find that indeed they seem to be completely different, and they seem to develop follow two different development trajectories, and that these GATA3, which is a transcription factor associated with Th2 response, there seems to also be important for the establishment of this this male-related Treg population, which so they show that depletion of GATA3 in Tregs really ablates this population, particularly in male, and that this results in metabolic dysregulation in males, but not so much in females. On the other hand, they show that in females, there's a less of these cells, so because they're not recruited by this IL-33 produced in males, but then they leave a niche of inflammation that produce, amongst other things, so it ends up having conventional T cells recruited to the fat, and that this production of, for example, interferon gamma is what ends up recruiting and transforming these Tregs that have this CXCR3 phenotype to the vat of female mice. So establishing, in a way, a regulation through slightly different mechanism, but in a way, both of these result in the recruitment of Tregs, but through different signaling, through different preferential signaling. And so whereas these GATA3 driven ST2 positive PPR gamma positive Tregs seem to be the most important in regulating glucose homeostasis in male mice, particularly, which are more kind of prone to having dysregulated glucose tolerance. In females, those cells are kind of competed out by the CXCR3 Th1 like Tregs, and that actually depleting this subset by, for example, having a TBET conditional knockout under the FOXP3 in FOXP3 positive cells actually results in improved glucose response in old females. So really showing that there's, kind of in a nutshell, what they show is that there's two. If you look at the visceral adipose tissue, there's Tregs that are preventing inflammation in that tissue, and that's very important. Inflammation in the visceral adipose tissue has negative effects in terms of glucose homeostasis, and also kind of general inflammatory homeostasis. In males, these Tregs are usually kind of developed as a response to IL-33 expression, which is preferentially done in males. And these are the Tregs that they take residence in the fat, and they keep inflammation at bay, and they help to maintain glucose tolerance. In females, this is less so. There's less IL-33 recruiting these Tregs or inducing their differentiation, and therefore this is mostly caused as a response of inflammation that occurs, and then these interferon gamma and Th1 responses are the ones that are recruiting or inducing the residence of these other different type of Tregs. But they're both kind of important for the normal functioning of the visceral adipose tissue.
So in other words, men get dad bod, and dad, which is fat in the belly, and that causes our Tregs to go wacky, and then we get insulin resistance, which leads to more dad bod.
In a way, what they show is that this high-fat diet reduces GATA3, this induction of this particular resident population by GATA3, and that is what it seems to be driving the loss of Tregs in the gut, and that is what gives you, and that is can be causing a glucose lower glucose regulation. So yeah.
So men, as they get older, just can't have fat.
Yeah, you gotta stay lean. So.
Well, that's depressing.
Yeah, or they don't do this in this paper, but it's been shown that apparently you can if you you can treat it IL-33 seems to specifically boost this population. I'm not sure if it's like topical or how you get it there, but you can use IL-33 in mice at least. IL-33 is administration seemed to help sustain this population in the vat. So maybe just do that. Inject yourself with IL-33. But yeah, I think the humans is not as clear. I think this mice system is a little bit more clean. But yeah, in general, the fact that we have T-regs helping us maintain you know homeostasis of our fat. Fat can be very inflammatory if left unchecked, for sure.
I wonder why we evolved this way.
Because I don't think because we just in principle we would never get so fat, right? I think we just never planned for it. Evolution never planned for us to be fat.
Well, I'm also going to talk about T-regs, but not so depressingly.
Oh,
I'm going to talk about how, in Nature Immunology paper, about how IL-23 stabilizes an effector T-reg cell program in the tumor microenvironment. So this first author is Tobias Wertheimer. Last authors are Sonia Tugase and Burkhard Becker. Came out on Valentine's Day. All right, so T-regs are bad, bad little children in cancer. The tumor microenvironment T-regs, as we know, are immunosuppressive and let cancer go wild. And this paper looks at one of the things causing it. And one of the things causing it is IL-23. I'm done now. No, so myeloid cells, myeloid cell-derived IL-23. In this paper, they look at it and they demonstrate that the myeloid cell IL-23 fuels tumor growth, which was known, but how it's doing it in part is by generating effector T-regs, which are immunosuppressive. And so that's the punchline. There's not a lot past that punchline other than they really demonstrate IL-23 through the IL-23 receptor on FOXP3 T-regs is generating immunosuppressive phenotype. But they do this with tissue-specific knockouts, with GFP, with YFP and GFP and RFP reporter strains and crosses and knocking things out. And they use the fact that I think the IL-23 receptor is on the X chromosome, so they can use natural mosaicism through X-linked inactivation to generate, natural mosaics. To demonstrate a bunch of this, and they hunt it all down and do way too much single-cell RNA-seq, just so much, to really look at all the profiles and validate, it's coming from myeloid cells. That's where the IL-23 is. These T-regs have different phenotypes. And with IL-23, they are this effector type, which is immunosuppressive. If you knock that IL-23 receptor down, the T-regs lose some of their FOXP3 and the levels go down. And then they produce more, inflammatory cytokines. And if you look at other T cells, they're producing more inflammatory cytokines. Without these T-regs, the T-regs come on board. There's less of it. But that's why you read the papers to go in all the gory detail. But fundamentally, they do a tour de force of cell line lineage tracing, essentially with these reporter strains. Not so much differentiation, but activation of effector status, right? From naive to effector status of T-regs, and really show that IL-23 in the tumor microenvironment, as derived from myeloid cells, leads to immunosuppression and growth of the tumor. If you get rid of it in these tumor models they did, they can kill the tumors then. The host takes over and the tumors shrink. And then similarly, they looked in human cells and saw there were better outcomes with less IL-23 in human cancers. They did a little bit of human work. Now, the good news is there's already an IL-23 antibody on the market. There's gazelkamab for psoriasis and IBD. So you already have an antibody that could be used in cancer based on this type of work, which means it's a really logical extension to go into human trials. So that's actually really cool, right? And these are used in IBD, and so you're worried about colon cancer risk with immunosuppression, but this suggests that in this case, you're not going to have that problem.
it's very interesting because when you think of IL-23, I think of more like as inducing Th17 cells. So, and I think there's also been some, there's we talked about in other papers about IL-6 and how important IL-6 receptor seems to be for Tregs in the muscle for muscle repair. So It always shows how much we underestimate how T-regs, they just do whatever they want, and they just take whatever signal they like, and they just will run with it and wreak havoc if they have to. Because these are bona fide T-regs, but they don't. They just get activated by sensing IL-23. So I thought it's very interesting. It's very remarkable.
Yeah. I mean, it's a really strong linkage that they go through, which is why it's worth reading the paper. But me describing all of it is they cross this mouse to that mouse and do a reporter thing and knock out one of the paths, then see this, and they do the other thing and trace that. I mean, it's
Yeah.
really just like every type of mouse combination you could possibly ever want. Flocks
Yeah.
is something else done to demonstrate a very robust linkage. So it's very well done. It's very strong work.
I think it is maybe I'm underestimating, but I think it's a bit controversial. So I think they probably had to really show their point, because I
Oh
think
yeah.
this is non-obvious in my when I think about it. So this is very interesting, because I would have not expected this.
No, which is kind of cool.
How long did they take for the revision? Do we have
Let's see here.
do we have the times?
Oh, good question. Let me look here. It's not on blast like it is for a Nature paper, right? This Nature Immunology, they tend to be more.
Yes. Yeah, I'm looking for it and I cannot find it. I'm very curious, because this also looks like a lot of work. I'm just like,
This is a lot of mouse breeding.
oh, well, actually, it's not. It's just a year.
So one round of here, go breed these mice, and then 6 months more work.
Yeah. Yeah. That's not too bad.
That's
Okay.
not the worst.
All right. Very, very interesting. I mean, these effector T-regs are like, it's hard because often effector T-regs are hard to culture and are hard to, so oftentimes we kind of ignore them, or they die, or we don't, it's hard sometimes, especially in this in vivo experiments, to follow up, follow those cells. So maybe that's why we didn't, I don't think we saw this coming. Okay. So for our last, paper today, just a quick, it's not gonna be a long paper, but I thought it was very interesting. It was published in Nature. Uh, and the title says it all. Smoking changes adaptive immunity with persistent effects. So kids don't smoke.
This
As I
is
said, published
tobacco, right?
tobacco smoking.
Got to be
Yes.
real specific with kids these days.
Oh, that's true. Yeah. I don't know about the rest, but definitely tobacco smoke has all those, the tar and all those inflammatory things. So just stay away from it. Um, so it was published in Nature, also on Valentine's Day. We have a lot of Valentine's papers today. Um, First author, Violaine Saint-André. I hope I didn't butcher the name. From the labs of Darek Duffy at the Institut Pasteur. And this is part of what they call this Milieu Intérieur Consortium, like the interior environment consortium. So in which it looks like they have like 1,000 people, which they have blood from, and they fill a questionnaire about lifestyle, and they try to find um, correlations between biological measurements and lifestyle. So what it looks to me is like, for this paper, basically what they look into is, they have all these I think it's 1,000 samples, and they try to identify correlations between uh, response to immunological stimuli and production of cytokines and certain proteins, just to see how it responds to activation, and then whether they can find correlations with lifestyle information. So basically, what they do is they have this whole blood stimulation, and they stimulate with different types of agonists, which can be roughly divided into either microbial products, BCG, which would be BCG or E. coli or Candida, either viruses. So they have influenza, infect with influenza, or with poly-IC, which is a DNA, so a nucleic acid related stimulus, and they have other T cell activators. So this enterotoxin B from Staph aureus, which is a super antigen, um, uh, or anti CD3, CD28, and or cytokines, TNF, I1 beta, interferon gamma, and then they measure in response to this after 20 hours of culture. They measure a variety of chemokines, cytokines, and other factors. And then basically they run kind of correlation with the things that I have in the files of these patients. And one of the things that really comes up and they focus on for this particular publication is the effect of smoking. So they see there are three elements that show a correlation with activation of cytokine production, kind of activation and response to this stimuli, and it's smoking, CMV status, and body mass index. So and CMV, I think we kind of know that CMV, especially if you're CMV positive, you're going to have mostly a slightly different repertoire of T cell. You're going to have more activated cells in your blood. You usually have an increase in this memory cells. So CNV is, I think, this already kind of been known and is fairly clear. I think also body mass index as well. But so they focus more on the smoking part because it's very interesting that they see that those patients that either smoke currently or when the sample was taken or there was so they show an increase in certain cytokines, inflammatory cytokines, and particularly they see a big signal when it comes to CXCL5 production after a stimulation with E. coli, more induction of IL-2 and IL-13 after stimulation with the super antigens that will stimulate preferentially T cells. And this also correlates with like previous smoking. So to some extent, past smokers also have some increased activation. And so they see activation. So kind of more adaptive immune system. So CXCL5, IL-2, IL-13. But they also see expression when you have innate immune stimulation. With I don't remember if it's with poly-IC, but they also seem to have some simulation for poly-IC and LPS, which are unspecific stimulation. They also seem that the smoking status really seems to correlate with response. And what they basically they look at it, and one of the things that they measure is DNA methylation changes to try to explain where this difference comes from. And they end up zeroing into some CpG sites and methylation sites that seem to be correlated specifically with smoking. And one of these, so they see so they do all these, fancy statistical analysis in order to kind of differentiate and pick up covariants that are really associated with smoking and not with the other uh, um, cofactors. But they identify a couple of sites. Some of them, for example, in the aryl hydrocarbon receptor repressor gene. So this is known, to be associated with inflammation. And they identify a couple of other genes that seem to be differentially methylated in smokers. And so this this results in a effect on the adaptive immune responses that is associated with this DNA methylation that acts as kind of uh, uh, transactivator of genes related to these activation, these immune responses. So I think that's kind of the most interesting finding. They also look at some other SNPs, but I think this is probably the nicest takeaway message from the paper, that they do seem to find specific methylation patterns that are associated with smoking, and that this affects the immune response in patients in response to specific stimuli, either to T cells or to innate cells.
I think it's interesting. It's so persistent after they stop smoking.
Yeah. Yeah. So they only see really the persistence for these effects on the T cells. So not so more on the innate, so on those innate signals, but it's mostly for the adaptive immune response that seems to be persistent, and kind of even related with the length of time since last smoking.
So if it's been 20 years, it's less than if it's been 2 years.
Yeah. Yeah.
Kids don't smoke.
No, it affects your epigenome. Just don't do it. It's not worth it.
Save an epigenome.
Exactly. So yeah, that's good that I don't smoke.
Yeah, I have never either. All right. Well, let's get going because it's going to be a fun part. We're going to be speaking here with Dr. James Allison, MD, Anderson Cancer Center in just a moment. But before we get to that, we're excited to announce the launch of a brand new podcast on mentorship and science. Brett and I are both hosts of several episodes of the Lab Coats and Life podcast. Find it at www.labcoatsandlifepodcast.com or wherever you get your podcasts. If you sign up for updates about the Lab Coats and Life podcast before March 27th, you'll be entered to a lab coat giveaway. Enter the contest at www.stemcell.com/labcoatcontest.
Today, we have a truly very special guest. I'm very excited about this conversation today that we're going to have with Dr. James Allison, the one and only. He is, of course, Regental Professor and Chair of the Department of Immunology at MD Anderson Cancer Center, and Executive Director of the Immunology Platform and Director of the James P. Allison Institute at the MD Anderson Cancer Center. He has been awarded the Nobel Prize in Medicine or Physiology in 2018 in recognition for his work on checkpoint inhibition. But he has had a very illustrious and long career that we are hoping to discuss with him today and to hear more about how he came about his work and any advice for younger scientists. So Professor Allison, thank you. It's such a great honor to have you here in the podcast. What a highlight. Thank you so much for making the time.
Oh, well, thank you. I'm glad to be here. I always love to talk about these things.
It's a pleasure to have you. All right, Brenda, I know you've been waiting. Fire away.
So I think everybody, your name by now is very well known amongst, the immunology community, and the impact of your work on checkpoint inhibition in patients and treatment of cancer, particularly, cancers such as melanoma, lung cancer is really indisputable. I think it's well known in the community. But I was hoping you can give us a quick recap of your kind of the start of your career and also some of your earlier work that is also very important, and then maybe build a little bit onto your current work and how you are continuing your research on T-cell immunology, checkpoint inhibition, cancer treatments. So maybe start a little bit with some of your early work on identifying the T-cell receptor, for example. Many might not be aware of that fact.
Yeah, I'd be happy to. As a kid, I was always interested in science and biology in particular. And when I got into the University of Texas at Austin, I majored in biology, but I was also interested in biochemistry. So I actually did most of my research on biochemical projects, particularly in protein chemistry, which was fun. But after a while, it gets a little bit repetitive. And I was lucky enough, I think it was my senior year as an undergraduate, actually, 1968, a long time ago, to have a lecture from a professor named Bill Mandy, who was an immunologist who taught a course in immunology, which at the time was mostly just about antibodies, immunoglobulins, and B cells, the kind of immune cell that makes antibodies. And there's quite a bit known. I mean, we know a lot more now, but there was quite a lot known about it. But it was interesting seeing the structure of the antibody molecule and how it, works to identify a lot of different things. But the last lecture of the semester, he gave a lecture on T cells, which are just being recognized as a real thing, and another component other than B cells. T for thymus, meaning they develop in the thymus gland, unlike B cells, which come directly come from bone marrow and birth. Anyway, it doesn't matter. But those are the two main types of cells. But while the antibodies is well known that that's the receptor, that's the thing that they use to recognize things. It was completely unknown in T cells, and his lecture basically said there was this new kind of cell that also it looks like the clones. You have millions and millions of different clones with different receptors, and they go through your body though, and they percolate through the tissues and through the blood and through the lymph and recognize virus-infected cells or potentially cancer cells and deal with them. And I just thought that is really cool. And so I went after class and said, how does that work? How do they recognize stuff? Is it with an antibody or, how do they, if it's not antibody, what do they do? How do they kill? How do they communicate with? And he said, nobody knows anything. He said, I really don't think there is such a thing. Actually, I think it's just a another kind of cell that's got an antibody molecule stuck on its surface or something. And I just thought, wow, this is so cool. And so at that point, I just decided I was going to switch or move from. I was doing some cancer-related projects in biochemistry because I had an interest in that due to my family history and extensive experience with that in my own family. And so someday I'd hope to do something about that. But T cells, I thought, well, this could be really fun. And so I then got a did a postdoc in an immunology lab. Unfortunately, I just got stuck doing protein purification to try to determine sequence of molecules called MHC, which are major histopathology, which are very important molecules and we know play a central role in the immune system. But the work itself was kind of boring because I was just purifying protein again and sequencing it. And so I I actually did a few things that I was actually not supposed to do, according to my PI. But anyway, I got my first faculty job based on some of that work at MD Anderson, although not at the main campus, at a new campus that just opened near Austin called the Science Park, which was a wonderful place. And um, my job there was partially to make antibodies to cancer cells and try to determine changes that occurred as a normal cell became a cancer cell. That was fun, except I was really interested in T cells. And so I decided that I was going to try to figure out what the antigen receptor was, because that was the holy grail in the early 80s. People had been looking for it since the 70s. And the literature, we didn't have a very extensive library. Remember, this before the internet. So I went into Austin on weekends and read the literature and said, this is just got to be crap, because there was contradictory stuff and stuff that made no sense to me, soluble factors, one soluble, one bound, and all this stuff. And finally, I just said, I'm going to jettison all that. I'm just going to take a completely agnostic approach to this and thinking as a biochemist and protein chemist what it ought to be. And then I'll look for such a molecule. And so I decided that it had to be a membrane bound thing. It was probably two chains that had a disulfide link thing, like building on antibodies, or maybe four chains. I didn't really know. But anyway, I used some biochemical tricks that I knew and showed that if you compared T cells and B cells purified and looked at the proteins on their surface, those that had disulfide bonds, which is just a chemical way of holding two different chains together, the only one on B cells really of any importance was the antibody molecule. We could show that. And there was this mysterious thing that looked just like I'd predicted on T cells and not on B cells. And so I then, got a lot of different clones of T cells in the form of T cell leukemias, reasoning that each one ought to have a different receptor, purified them, sequenced them, and showed that they had constant and variable regions. And it was exactly what you'd expect the T cell receptor to be. It was nothing like anybody else had published before. And since I was a guy at this little place nobody had heard of, and I'd never been heard of, I had a hard time getting anybody to pay attention to it at meetings and things. But as what happened, Pippa Merrick, who was really one of the leading T cell biologists at the time, had made a monoclonal antibody. One of her students read my paper and used their antibody to purify the thing that activated those cells. And it was the same structure. And then I sent them some of the antibodies that I'd made and showed that it reacted with the receptor they had on a T cell hybridoma that actually activated or blocked it, depending on what you did. And it was the formal proof that was correct. And then we tried to clone the genes, again, using more of a two approaches, but Mark Davis got there before we did. And so, that's history. But, we at least were able to tell him after he got there was an acidic chain and a basic chain. And we showed they both, had constant variable regions. So he got the beta chain first. And at least everybody knew from our work, there was still some work to be done. And so, later on, people got the gamma, and then finally the alpha chain, and the rest is history. But that was a lot of fun. It was done. We did it strictly by a biochemical approach, and then others provided the functional link that really prove that. But I really wanted to keep going, because that's just the ignition switch, is all that is. And knew it got the T cell regulation had to be more complicated. But that work attracted the attention of the immunology community. I went to Stanford for almost a year and worked in Herb Weissman's lab trying to do the cloning. Again, of course, we lost that race to Mark and to Tack. But it did work out. I mean, I learned some molecular biology, but it also got me. I was asked to give a seminar at Berkeley, University of California at Berkeley, and I gave a talk, and they offered me a job. It was fortuitous. I was an assistant professor at UT, and they offered me a job as full professor. So I moved there and expanded the work and went on to show that people knew that it was more complicated than just that molecule. The cellular immunologists knew that there was what was called a co-stimulatory factor, an obligatory second signal. And so we did a lot of work showing that the molecule called CD28, another molecule of was that molecule, was the co-stimulatory molecule, which is kind of like the brake, the accelerator pedal, of the process. But when we got that molecule, there was a gene that had already been cloned, and we sequenced it. There was a gene that had already been cloned by a French group that was just trying to clone everything that they thought was T cell specific. And one of them, I think it was called CTLA4, cytotoxic T lymphocyte antigen associated 4, and quite a mouthful. But turned out it wasn't T cell associated. Its function was completely unknown. But what caught our attention was that it was highly homologous to CD28, at least on parts of the outside domain, which would be what you would expect to recognize things. But the inside was different. And any event, so we made antibodies to that, cloned it, and everybody was worried about that, was concerned with that molecule. We weren't the first. Another group got it and said it's another CD28, which made sense since it was highly homologous. But we made an antibody in our own work. Max Krummel in the lab at the time. One of the things we realized, if you have T cells or anything, and you put them in a dish and you give them some signals, and all of a sudden they do more of whatever they're doing, they divide faster, they make more lymphokines or whatever, that it's probably a positive signal. But if that's all you do, and you've got other cells there, you don't know whether you're giving a positive signal or removing a negative. And so the only experiment that the other guys did was one that would show that you got more, interleukin-2 or whatever. But of course, their experiment didn't at all rule out there was a negative one. And so we realized, you can't just stop when you're doing the experiment that gives you the answer that you expected. Is relatively easy, but that's not the game. The game is to do an experiment that tells you that you're right, that it can't be anything else. And that was the key thing that led us in that work. I mean, we weren't the first to get it, but we said, well, they only did half the work. We got to do an experiment that proves that it's a go signal instead of removing an off signal. And so we did some extra experiments, which I won't go into, but had to do with cross-linking it instead of having it in soluble form, and did it 2 or 3 different ways and said, no, it's an off signal. And you're just blocking an off signal, which is why it's like that. And that was really controversial for a while. But my main advice to scientists is really think about that when you get data. It's not good enough to get data that's consistent with your hypothesis. It needs to rule out the contrary hypothesis. If there are two, and you got to always remember, just think about it. Look at the data, think about it from every angle, and then say, is there something else that can do that we're missing? And so anyway, that was the key to that. And so we showed it was a negative, and that was made for a grist of discussions at Gordon conferences and these very elite scientific conferences for a couple of years, where the textbooks even had it in as a co-stimulatory molecule. So we decided, Jeff Bluestone was another immunologist that came to the same conclusion as we did. And so, ultimately, I think we were proven right, mostly by the knockouts that came along. They were made by Tech Mac and my lab and Rolling Sharp. But again, the knockout was consistent with it, but didn't really prove it. I have to say that. They weren't as knockouts usually considered to be. But in the meantime, one of the things that occurred to me, because I had this interest in cancer, was maybe this tells us something about why vaccines haven't worked, because we knew that by studying regulation of CTLA-4 production, we realized that it's driven by the T cell receptor signal. It's not there on a resting T cell, but after it gets a TCR signal, it starts making it. And we showed quantitatively every time it gets another T cell receptor signal, it makes more. And so I started thinking, well, maybe one of the reasons all these tumor vaccine trials have failed isn't because the antigen, the vaccine that they're using is the wrong one, or as many people thought, the immune system just can't see tumor cells because they're too close to normal. Maybe it's just because they're giving the off signal. And so first experiments we did to just determine whether there was a lack of antigens, which is what a lot of people thought, is we put the ligands, other people had discovered the ligands for CD28 and CTLA-4 molecules called B7. And we took tumors that grew in mice and would kill them. All we need to do is put a B7 molecule that gave the CD28 signal, and they'd be rejected. And so they had the antigens. The reason they're not recognized is because they didn't have co-stimulation. They didn't push the gas pedal. They gave the ignition switch signal, but not the gas pedal. And so they're effectively invisible. And so once we realized that, then I thought, well, what happens then is that CTLA-4 might be coming along. And because the T cells can't see the antigen until they get picked up by cells that can give that B7 signal, and which was known to be death and inflammation. You have to get that first. So the tumor has a head start. And then maybe if they get too big, then CTLA-4 stops the expansion of T cells before you can get a sufficient number to kill them. And so the obvious experiment was to see if we block CTLA-4 in vivo where there's a tumor, could we get it projected? And sure enough, that with every tumor model we looked at, either by itself or with a vaccine or with some chemotherapy or with radiation, we could cure those tumors. And so that's kind of how we went from just learning the basic mechanisms of T cell regulation, working out that, the antigen receptor wasn't the whole story. There was a gas pedal and a brake. And once you understand those, you can start thinking about how to, treat cancer. And so
You had a couple really interesting insights you mentioned. One was looking at not just your results and, it matches what I expect, but making sure it doesn't exclude other possibilities. And that's really insight. The other thing you talked about is you took an agnostic physical or biochemistry approach, which I like. I'm a biochemist by training as well. I was wondering if you could talk a little bit about that general approach to biology, that, that structural, agnostic, mechanistic, physics approach, as it were, because chemistry and biochemistry and physics are all, almost the same in a way, and how that has helped drive decisions. I see it in other parts of science as well.
Yeah, I think it's particularly useful when there's a lot of confusing data out there, and the obvious thing, or not so obvious, but I think people have taken other approaches and just gotten nowhere, and there's a lot of contradictory data. I mean, that's at least the way I felt about it. I went into the thing thinking, I would learn from reading literature, but all I did was get confused. So, but I think in a situation like that, that's the best way to do it. Just back off, say, I'm not going to have my mind cluttered with a bunch of data that I don't understand, and it seems to be confusing and self-contradictory. I want to just clean it up, by reasoning what I know based on just the minimum things that I know, what it should be like, and test that instead of just getting a factor, and making stories up about it, which is what I think a lot of people did. But anyway, I think it's a valid approach, particularly, as I said, when there's a lot of confusion around, and making any assumption based on that work is going to lead you down the wrong path.
And so talking about assumptions and expectations and how this relates to your to this work and the discovery of CTLA-4. So you kind of touched upon that, but I think when I hear it's hard to remember these things that we take for granted. They didn't exist before. And so, and you mentioned that it was kind of controversial to think about um, T cells having an off switch, an on switch, a break. How has our understanding from the start evolved about how do T cells see around them? How do they respond? Was it surprising when you think about this a second signal? How did people start realizing that a second signal was necessary? What are kind of experiments that guided our early understanding of T cell activation?
Yeah, the early experiments were actually done by Ron Swartz and Mark Jenkins at the National Institutes of Health, when people knew that these cells called dendritic cells were really the best cells that, both activating a naive T cell, and there was a phenomena called anergy, where if a T cell just got an antigen receptor signal, they could be turned off. But what they showed was that you could have the right antigen. Anyways, what you have to know is that T cells recognize antigens in the context of major histocompatibility antigens. So there's actually two structures that are recognized, a bit of a peptide in the context of the protein. But if you have the right antigen, but the wrong MHC molecule, T cells don't get activated. But if you have the right antigen and you inactivate the co-stimulatory signal, the B7 thing, they still don't get activated. Sorry, you have the right antigen and the right MHC, but you don't have this other thing. That's not sufficient to activate. People showed you could chemically knock that out and then add back another dendritic cell that had the wrong MHC and couldn't interact with a T cell receptor at all, and that would be sufficient to restore it. And so that was the observation. So the original definition of co-stimulation was an obligatory second signal that T cells need in addition to that antigen receptor engagement. But as we realized, while I think that CD28 is still the only molecule that matches that, people began to realize there were a lot of other molecules that would add to that or inhibit it. In some cases, there were both types found. They're still now they're all called co-stimulatory or co-inhibitory. And what we realize is that it's a lot more complicated than we thought. We don't know the number of those yet. It probably numbers in, I would say, a dozen or so molecules that potentially have that sort. And depending on the beautiful thing about the immune system is it does a lot of different functions depending on whether you've got a bacterial infection, a virus infection, a cancer cell, whatever, it responds in different ways. And you tuned with different cytokines being produced. And so there, all these other molecules play a role in regulating how that plays out. And we're still figuring that out. There's another type of cell called the macrophages is a main type of cell there, but there are really a lot of different types of these cells that can either activate or turn off T cells, and there's some molecules on those too that can shape an immune response. And so what now we realize it's not just on or off, it's the quality of the response is also shaped by all those signals. And these myeloid cells, of which macrophages are a type, are a really complicated set of cells that may differ by only 2 or 3 genes that are being expressed, but as a consequence of that, have very different effects on T cells. And it's understanding that those cells are almost like a continuum. It's hard to say there's one type or two types or three types. And we realize that T cells too are somewhat plastic in their function, that adapt to what's happening when the immune response is initiated and things that can shape it as it rolls out. And so just to make sure you get the right response at the right place at the right time and don't cause autoimmunity. And the number of these entities is very, very, I'd say it's a dozen or so now, at least, that are being looked at. And so it's very complicated. It's one of the beauties of biology. We think we've got it figured out, and then we understand that we've seen the prototype, and biology is more of a continuum than discrete boxes, and we need to not try to put things in boxes and say this is this kind of thing, because they'll blend into each other now when you get enough data and realize what's going on.
That sounds like my smear hypothesis of T cells I keep telling Brenda about.
Well, that's what myeloid cells in particular look like a smear. If you look at single cell RNA, you see, and you group them by the modern computational things, group them according to who's more similar, who's more different. It does look like a smear, just a big blob on a graph.
Yeah. So CTLA4, you figured out that there was a inhibitory stimulus. What was the journey to the clinic, which is where, it's had such a profound impact? I mean, I'm sure it was rapidly adopted and immediately moved in with no result.
Well, yeah, right. So we got the result, and it was, like I said, it was while the debate was still going on. We already had the cancer data when people were still arguing whether CTLA4 was a positive or a negative. And I think that our data, together with the knockouts, was the strongest evidence that it really was an inhibitory signal. We showed when you took it away, tumors got rejected. But anyway, so I carefully told the patent, the UC patent office, I want to do this right so they get patent. So I'm going to talk about it at the International Symposium, or International Association of Immunology Societies in San Francisco in '95. And so I presented it there, and everybody went, "Oh, cool," and that was about it. And at the time, the genome sequencing was quite popular, and it became evident that cancer was caused by driver mutations, locking on signals that normally would be off and telling a cell to divide, for example. And because of the failure of all the vaccine trials, immunology had just fallen in disfavor. And so I went to, I don't even remember how many, big pharma, little pharma startups. And, they just said, no, immunotherapy will never work. And for over 4 years after that paper was published, it took to find it. And it was really infuriating time, because people would say, well, it'll never work. And I said, you're a scientist. How can you say that? Look at the data. If you want to tell me it's not going to work, tell me something's wrong with my data. Look at my data. They say, well, it'll just never work in humans. It's a mouse thing, that is to say, it never has, so it never will, and it works in mice, but it'll never work. You're not going to know unless you try. But think of it, if it worked, we can cure all cancers, because you're modulating the immune system, not the cancer cell. You're not attacking the cancer cell. So the kind of cancer wouldn't matter, and still. And finally, the only way it worked was there was a company that called Metarex that had access to mice that had the antibody genes replaced by humans, and provided a very quick and easy way to a clinical reagent. Only they didn't have any clients. And so we had a perfect test for them, and we teamed up, and actually with a friend named Alan Corman, and I really started making the first antibody, making the immunogen to use, the human chimeric protein. And then Niels Longberg was the head of this company. And so they made first antibody, MDX010, it's called, became ipilimumab. But so finally it went into early clinical trials, and they were done, some of the early ones were done in metastatic melanoma, which at the time had the median survival was 7 months after diagnosis, and fewer than 3% of people were alive at 5 years. And yet there were, I think it was 3 out of 14 patients in the safety trial. One injection of antibody, the tumors all went away. And some of those were still alive 20 years later. Anyway, it went into early trials, and we know that ipilimumab by itself cures about 20% of patients with metastatic melanoma. And later on, PD-1 came along from Hanjo and Arlene Sharp and Gordon Freeman. It hits another checkpoint that works on at a different point, not during priming of a T cell like CTLA-4 works, but works on T cells that are fully differentiated and inhibits their function. So it also works against, and these work against many different kinds of tumors. But because of the different way in which they work, of course, you can put them together. And right now, the two of those antibodies, there's a randomized trial, very large randomized trial, which is the gold standard, in clinical studies. That's 8.5 years out now, with over 50% of the patients still alive. So now, and so 8 years, well, actually, it's more closer to 9 now, maybe even 10. But anyway, that's considered curative. There's one of the patients that I met, was treated in 2004. At the time, she has 2 kids now, still doing great. So these drugs are curative on a fraction of patients. And but anyway, so it went from nobody would believe it to that first result in melanoma. And then everybody said, "Oh, this is a great melanoma drug," but it's because they have so many mutations and so many targets. And then because for example, some types of lung cancer have the same thing. Lung cancer responds very well. But now we know that some cancers, like kidney cancer, that don't have very many mutations, also respond very well. And anyway, so it's went from nobody believed it to everybody now uses it for everything. And I mean, the good news is it works about 20% of the time. That's also the bad news, though, of course, because, but I think that the job now is to really get into the other cells in that smear of those cells that interact and regulate T cells and find out how to deal with those. And I think we'll get it up. And also to try different ways of doing it. There have been some trials recently of combinations or single antibodies even, in what's called neoadjuvant trials, where you start giving the drug and then you do surgery and then continue the drug, that have response rates over 90% in lung cancer and in melanoma. I mean, these are too early to say how durable they're going to be, but there's no reason to expect that they won't be as durable. So now everybody, wants it. Everybody wants their drug. Unfortunately, it's field's gotten so crowded. They're doing many combinations without any data. If I could go on a soapbox for a minute, what now is companies are combining everything they own, or maybe making some things without really thinking about the biology. And so there are these 100 patient trials. There are over 3,000 trials of 50 to 100 patient trials. Look, that take 3 to 5 years. You look for a statistical difference, and we know now from those patients that where it's successful, we know what the signature for success is. Mm. And so what needs to be done is what we're trying to pioneer now. If I could give a brief commercial for our institute, is really do small trials, something pioneered by Pam Sharma here, where you treat a dozen patients, perhaps. You get biopsies of the sample very early after therapy and look at what happened. What are the cells? How they change? What are you missing? Because we know what the success looks like. What are you missing? Is there some type of cell there that we know is inhibitory? Did a new molecule pop up? Because some of these inhibitory molecules I was mentioning earlier, some of them pop up in prostate cancer, but not melanoma. Others pop up in melanoma, but not prostate cancer. So it's more complicated than we understand. But we need to get those data because, if you've got an antibody to one of these molecules and you combine it, that combination may be great for prostate cancer, but if you test it in melanoma, it's not going to work. And then you'll say it doesn't work and throw it in the trash and nobody will ever visit it again, So we're killing a lot of things that have potential by not looking at the mechanism early on. And so I think that's one of the pushes that we're trying to make is get away from these 500, 100 to 500 or whatever, these large trials where you just use statistics to do small trials to guide them, de-risk them, and come up with a combination that works before you even look for a clinical signal.
So, regarding CTLA-4 inhibition, now that we have the expert on the show, I have a couple of questions regarding what do we understand exactly about the mechanism of action in cancer patients? Because of course, inhibition of the inhibitor in effector cells is a great part, but there's also some data suggesting that uh, depletion of T-regs might be involved, and there's these differences between different companies are making different CTLA-4 inhibitors, and somehow they don't work exactly the same. So what do we understand about that now?
No, you're exactly right. What we showed in mice was that there's a cell intrinsic mechanism. I mean, our work was done we did it before regulatory T cells had been recognized, but we did it with purified cells where there weren't any of those around. But now we know that some antibodies do deplete those. It's a secondary mechanism because regulatory T cells express tons and tons of CTLA-4, which gets so dense that that could work in a technique where certain macrophages will kill them if they're coated with antibody. So, so some of the antibodies that are clinically used can do that. Um, and so, but CTLA-4, the first one, CTLA-4, does not do that. It does not deplete. Some of our mouse antibodies do, some of them don't. And the ones that do deplete tend to work better than the ones that don't. So I guess the bottom line on that was there's still room to improve ipilimumab, either with a separate way of killing regulatory T cells or by engineering it, or it will do that as well. The danger of that is that you increase the chance of adverse events as well, autoimmunity and things. So, with all this stuff, there's a balance, but that is a good point. The mechanism is complex depending on the very nature of the reagent that's being used. Both the antibodies that are against CTLA-4 that are in common usage now, both of those, as I should say, neither of those depletes T-regs. And so there's a lot more could be gained if we could do that as well. But as I said, the trade-off would be most likely additional toxicity.
Which is not minor, no? Because CTLA-4
Yeah, it can be major. In some cases, it can be lethal.
Yeah.
But now people know how to catch it early, and not always, but most of the times it can be managed with immunosuppressive agents, which don't get in the way of the tumor, any tumor effect.
So to go with that, you talked a little about the future, right? With these small trials that really get some mechanism, which I empathize with from my day job. It's a similar approach because sometimes you just got to get into people and understand. What else do you think is next in the immunotherapy land? You mentioned like some, cancer specific inhibitory signals. So I'll put that as one thing you've mentioned. Is there anything else that you're kind of looking forward, or that 360 view you like to take where you kind of try to see the problem for all sides? You see some other holes in our understanding you really want people to go after?
Well, I think these myeloid cells. I mean, one of the big problems in glioblastoma and pancreatic cancer, I mean, there's some different ones, but one of the things that they share are very high levels of suppressive myeloid cells that we've got to break through somehow to get it work. But some work that we've done and others, but we have a paper in press showing that if you do the treatment properly, you can turn the suppressive myeloid cells into ones that support T cell killing and can add to it. And so I think that's one of the next fields is figuring out, forget the T cells, but finding ways of epigenetically shifting the myeloid cells from a suppressive to a more inflammatory phenotype that would support killing tumors. A whole new frontier, I think, of understanding, because it's clear that these are, not to get too complicated, but these are ways of changing gene expression by changing certain signaling pathways in the in the cells, the genes that are made to change their function. We're just beginning to learn how to do that.
Yeah, I guess the T cells can only go where the myeloid cells are taking them in a way. So they need the right instruction. And so there's so much that has been done. So you mentioned so many clinical trials, so much change in the kind of mind shift on our understanding on cancer and what we can do about it. I think immunotherapy of cancer, a lot of these. I'm a big fan, and I'm always saying I used to work in an institute that did extensive checkpoint inhibition trials, and it's just fascinating to see how all of a sudden we're not thinking about killing the cancer cell, putting chemotherapy, radiation, but actually getting your own immune system to do it. How, from a more kind of a personal point of view, how does it feel to know that your work has been so instrumental in this huge kind of earthquake in our in treatment? And what is the kind of satisfaction that you get from not being here and having done this?
Well, a lot. I mean, as I mentioned earlier, I watched my mother passed away from lymphoma, and she had burns on her neck from radiation, and her brother died from lung cancer, and he went from being a robust Marlboro-like man. When he was a cowboy, he ran a ranch, and to just a skeleton almost in an oxygen tent because all the chemotherapy that he had gotten, and another uncle that died because he had melanoma and there wasn't any treatment. So he just said, hell with it. I'm just going to enjoy my life, for the last few months. And I said, there's got to be a better way. I thought that a long time ago. And I'm just glad to have had a chance to do that. And I tell you, every time I meet a patient, that has benefited, I don't know, it just affects me in an emotional way that's hard to describe. I'm just so just glad that I had played a role in helping people like that. I mean, it feels pretty good. I mean, it's all the awards and all that stuff is one thing, but just meeting, seeing people that are alive because of the treatments is the reward. And but as I told you earlier, we're only getting about 20% of all cancers now, so there's still work to do. So.
Yeah, for sure. The work is never done, but definitely a huge improvement. And so many people, owe their lives to all this new technologies. It's just great. I always fascinated. I'm always so grateful to be, you know, witnessing all of this.
So, in an alternative reality where you weren't saving so many lives by going into science and biology and doing the work you've done, what would you be doing?
Well, I think I'd like to be a musician. I mean, I play harmonica, blues harmonica a bit, country western and blues, and I've played with a number of you know bands, garage type bands over the year, including now I play with a group called the Checkpoints. Oddly enough, made up of cancer scientists, and one of the really fun things about that is a little bit like the lab in that to do science now, and even back before everything got as specialized as it is now, different people in the lab have different skills, and putting together, people's different ways of looking at things and putting together with it all together, and and you feel like we got a team, we're going, da da da, everybody contributes. It's a lot like getting a band together, when everybody's in tune, or not, maybe not too well in tune, but anyway, get together enough to really have that when it clicks is really fun. And I sit in with Willie Nelson quite a bit when he comes to Houston, and had the had the fun of. I mean, he tolerates me on the stage for a while, mainly because I'm a good friend of his. Mickey Raphael, Raphael, his regular harmonica player, and so Mickey lets me come out every do a few songs when he's in town, and that's quite a thrill too. So just the fun of playing in front of people is a lot. But the other part is that it's a lot like having a lab.
For those listeners who don't know, you often delight Congress attendees with your skills and your band. So you did play it at the IOS conference, was it?
Yeah, we played there. Well, that actually was we just got together a bunch of people. Two of us, Tom Gajewski and I, had played together before, but none of the rest of them had. But the common thing was music, and we
Yeah.
managed to even do what was it, "Miss You." We did a Stone song, "Miss You," that I'd never played before, and it sounded okay.
I'm sure people either liked it, didn't mind, or don't remember. So
Yeah.
this has been such a wonderful conversation. It's been such a pleasure to talk to you.
Yeah.
We could get going for a long time, but I think that we are reaching the end of our time here. But so thank you so much for joining us. Of course, James Allison, to the Immunology podcast. I'm sure our listeners really enjoyed this conversation. And we're looking forward to see what's next for you from your lab, from your collaborators, and what's next in immunotherapy. It's just a magical field, I would say.
Thank you.
Well, that brings us to the end of the show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup papers. You can reach out to us on X, aka Twitter, at Immunopodcast, or via email at info@immunologypodcast.com with feedback, or if you want to suggest a guest.